Okay, let's talk about the mathematical frameworks, because we are going to talk about more than one framework for drug concentration dynamics. So this lecture is also like a conceptual introduction to the common mathematical modeling types that I can say in this way. So last time we discussed that in pharmacokinetics, we are interested in the temporal distribution of drug concentration in the plasma. So we are interested in, so wanted to know. Want to know Z total is a function of time. So after we knowing this, so what? That's a realistic question. Like, why do we ever solve for build a model to solve for the concentration of drug in the plasma? This is because a lot of physiological responses to drug depends on the drug concentration. So this is because physiological responses to drugs depend on C total. So what do we mean physiological responses? It can be like body temperature, body pressure, heart rate, plasma glucose concentration, cholesterol level. So for example, after someone take a drug, like a pill of drug, and then they wanted to know, okay, how does body temperature change with after taking the drug? Or how does body pressure changes with after taking the drug? That's the meaning of physiological responses. And that's determined on the concentration, of course. So there is a sort of a relation, which is also like a sigmoid relation. This is more like similar to Hill equations method. So the physical response to drug depending on so the response. What do we call it? We call it response function, response equation, response equation. R means response. R drug equals to R maximum multiplied by the total concentration of drug to the power of n. N is a Hill's constant. C50 to the power of n plus C total n. So here, n is a Hill coefficient. So n is, we can call the Hill constant Hill coefficient. It's the same thing. And now who do you talk to? Some people you would like to use coefficient, some people would like to use constant. It's okay. And R is the maximal attainable response. So R maximal is the maximal attainable response. And the C50 is the drug concentration at which the response is half maximal. It's also makes sense. We have seen this in oxygen concentration. We have like P50. It's the same way we define things. So this is the drug concentration. at which the response is half maximal. So the derivation of this model, we are not going to derive it here. You can imagine that it's very similar to the Hill's equation setup when we discuss about the hemoglobin and oxygen. So you can think about like a drug being like as a ligand and they have a receptor at the cell surface or on the cell membrane. So it's a ligand receptor binding model. Then you can build up like a chemistry model, like you have a ligand and a receptor being separated, and then they can form a combined form, and then this process can go back and forth, and then you will have exactly the same scenario, and you can derive this. So this is why we wanted to know C total, because it's related to the physiological response in the body. And then in order to get the C total, there are a lot of parameters involved. And it is theoretically, if you want to like build a theoretical model, you need some modeling mechanisms or strategies. So this here, we are going to talk about the three common mathematical frameworks. The first is called compartmental models. The idea of compartmental models is, as you can see, compartment means you divided the human body into several compartments. For example, you can buy, just for example, by spatial organization, just by spatial organization. It's not a good way to say it, but you will get what I mean. So by spatial organization means, okay, you model this as a one compartment. This is another compartment. This is another compartment. Each rectangular is a compartment. So then you may have like 1, 2, 3, 4, 5, 6, 7. You have 7 compartments. And within each compartment, the concentration considered as the same. And the flux, the mass transfer is between compartments. You probably recognize this may not be a good way to compartmentalize the body because body depends on what you are modeling. Some biomechanics, maybe it's reasonable to separate this way. Sometimes maybe it's better to compartmentalize by organs or tissues. For example, you model the heart as one compartment, and you model the lung as another compartment, and you model the stomach as another compartment, and you model the intestine. No, this isn't look like the intestine. You model the intestine as another compartment. And then you build the links between these different compartments. And then the transport is between compartments. So there are different ways you can compartmentalize the body depending on your needs and depending on what makes sense. So the idea is the drug concentration in each compartment In each compartment, concentrations in each compartment are considered the same. Maybe it's not a good grammar. What I mean is, if you, for example, you have a lung, you have kidney, you have intestine, then each compartment got its own concentration, and the concentration in its entire organ remain the same. That means we don't further, for example, we don't further divide um the stomach into different sections, and then we are saying, okay, section one get one concentration, section two get another concentration. No, we don't do that. We're just saying each organ is the entire thing, and the concentration in each organ is the same. But the different compartment is in general have different concentrations. So the drug concentration, so sorry, the drug in this model we consider drug transport across different compartments. So that's the idea, general idea of compartmental models. And for the study of um, pharmacokinetics, we will mostly use compartmental models. Okay. Well, you can see compartmental models. They take a lot of simplifications because they just lump each compartment as a unit. So these are also called lumped models. Those extremely simplified models provide some practical like simplification for a lot of scenarios, and that's why they become popular. And it also makes sense in a lot of scenarios. The second type of model is called the physiological models. So those models, by the way, the lumped models, or you can call it lumped models or well mixed models. Well mixed means within each compartment they assume like the solute, like if you take a drug as a solute, the drug is well mixed within the entire solution. So if you think like the entire like intestine as an organ, it is a big system, it's a big solution of stuffs. And it's so well mixed that every part of it get the same concentration. For physiological models, we don't assume well mixed. We usually consider spatial variations, consider temporal and spatial variations, and also follow physical laws. So you usually consider spatial temporal variations, and they also usually build the models that build on first principles. Physics principles. Such as conservation momentum, conservation mass, F equals ma, these type of things. And you usually need to take differentiation forms. And you need some like differential equation. Usually equation in many cases. So you may have other features. So this is a more nuanced model. The models, mathematical models will have been studied in the previous, like a good number of lectures, almost the entire course up to this point. What we talked before, like filtrations, fixed laws, fluid flows, boundary layers, like like porosities, diffusion coefficient, these type of things that they all belong to these physiological models, this category. And another example is it's the Burger cylinder model. If you recall it, we developed a Coxeter model of oxygen. And then those models, as you have been experienced so far, they need some sometimes they need some special treatment. And in many cases, there are no analytical solutions, and you need to like make simplifications to assume certain geometry or use numerical methods or other methods. So they are more involved, but also like more physiologically descriptive. Maybe that's another way to say it. So the third type of model, we can give it a nice looking name. It's called a model independent approaches. Model independent approaches. Okay, this is a nice name. If you want to give it a not so nice professional name, well, it's still professional. If you want to give it another name, basically it's just data fitting. So it means you don't invoke any modeling. It's fitting. For example, for an experiment, you get something, you obtain something as something x as a function of time, whatever you are going to measure. And then maybe you got this curve, whatever. And you don't know what's the governing equations behind that, and you don't maybe you also you either don't know or you don't care or whatever. You don't. The result is you don't write out an equation to describe the physiological process or whatever. But what you do is, okay, let's treat it as a field data fitting. Maybe I can fit a data look like this. Then you write the expression of this curve fitted line, and you will give it an equation. This equation would be your modeling result. So basically, it's data fitting. So a common way to filter data is like if it's linear, then it's good. It's not. If it's nonlinear, is it quadratic or cubic or exponential or logarithm? Sometimes if you see a nonlinear curve and you can take a log, and then you see, do they follow a linear line after you take a logarithm? And that may give you something, maybe they are exponential or exponent functions. So you can use various like data fitting tools. And this is model independent. We are not going to do this because this is not like a data science class or data fitting class. I'm just telling you that in a case you don't know any, or you don't want to model anything, you can still fit a curve. And it's not that bad thing, because sometimes there are also positive things about data fitting, because once you fit it, you may say, okay, I get exponent value of n equals 2, but instead of 3. Then those, or I get a slope, a high slope, a low slope, a steep, and a flat, or whatever. Those fitting, sometimes they actually can give you hints of the underlying biophysical mechanisms that sometimes it may be harder to obtain from first principle, or sometimes there even no such first principle ever exist. So data fitting has its good thing. It's just not a common way of standard modeling. It's considered as you use the result of data fitting to get insights of what may going on, and that's another way to look at. Okay, so let me leave a homework for you. So to consider the drug response function given by this one is the same as we discussed earlier. Let the desired drug effect to be a fixed proportion alpha of the maximal response. So that means R drug equals to like alpha R maximal. Use this alpha to derive an explicit expression for the drug concentration T total in terms of alpha, C50, and N. It's just a rearranged equation. It's just solve an algebraic equation. And this exercise is meant to give you some hands-on experience and some feeling of manipulating equations. That those type of experience will help you develop intuitions and those type of understanding you may need in the future.